Hello, my name is Dr. Shanda Blackmon. Welcome to the Baylor College of Medicine Lung Institute podcast series. We're here to communicate with all of our care providers, our learners, and our community, as well as patients, about how to improve lung health, how to educate you and all of the people around you about ways to prevent lung disease, treat lung disease, and maintain lung health. Hello, and welcome to the Baylor College of Medicine on-air Lung Institute podcast. I'm so excited about our guest today, Dr. Richard Gibbs. He leads our Human Genome Sequencing program and has historically been at the forefront of genomic research for decades. We're so excited to have him here. He's a key player in the Lung Institute. Welcome, Dr. Gibbs. Great to see you.
Thank you very much.
Can I call you Richard?
Please do.
Great. So tell us a little bit about your program, how you came to Baylor, and everything that's happened. I know about it, but I want all of our listeners to hear about it.
Well, thank you. I'll give you the quick history.
Okay.
Going all the way back to the early 1990s, actually a little before that, when the Human Genome Project was conceived. And we were working in the general area of genomics and developing technologies for sequencing genes and so forth. And so as the program ramped up, we participated, grew, and eventually became one of the 5 centres that actually completed the Human Genome Project. And the reason that we were doing that really was to enhance our genetic studies. So we were, or are, geneticists who recognise that genomics empowers genetics, and sequencing the human genome was really the basic platform that we needed to really, put a rocket behind human genetics. So we built the Human Genome Sequencing Center here at Baylor College of Medicine to sequence the first human genome, and then that was completed in 2003. There was a draft in 2000 that I think most people associate with the completion of the project, but it actually went to 2003 when we had a essentially complete genome. But since then, we've gone on to do more genetics,
right,
and to hopefully translate genomic technologies into the clinic, which is one of the reasons that I'm so very excited that you've formed the Lung Institute and that we're here talking today.
Yeah, so I remember coming to Baylor College of Medicine as a resident in 2003, the year that you finally completed the sequencing of the human genome, and you were the talk of the town. It was very exciting to hear about all of the technology that you were enabling us to use as clinicians. I think the big question that we had back then was, how will they organize all of this information? It's just an unsurmountable, inconceivable amount of data and information. And it seemed to me that the science of sequencing the human genome was just as complex as the science of analyzing it and trying to make sense of it. And so we saw 2 different specialties evolving from this. One specialty generating the data and one specialty making sense of the data. Is that a nice summary of?
I think you really described it very well. In fact, the challenge of interpretation and certainly of functional understanding, we're really just at the beginning of that journey. It's the immensity of all of medical biology.
Yeah, and then back then we talked about sequencing genetic data, and now we talk about whole genome exome sequencing. We talk about proteins, genomes. Tell our audience a little bit about how we look at that. What is the difference between whole genome exome sequencing versus just looking for specific mutations, fusions, deletions, alterations?
Yeah, I think the way to understand that best is to think about how big the human genome is, how immense it is. There are 3 billion DNA letters that you get from each of your parents, so you have 6 billion letters. And any one of those can actually trigger disease, fully penetrant genetic disease. So to do the job properly, if you want to do a genetic analysis on an individual, you really have to ascertain all of those 6 billion bases precisely, and then have some way to figure out which ones might not match what you expected, and then what the consequences of that are. And of course, this is the complexity of this is enhanced by our genetic diversity. So each new person you sequence will have about 3 million differences with the previous database sequence. And so, each of us are very different at the level of rare variation. So basically what that means, if someone walks into your clinic with their sequence and you look at genes, even genes that you know very well, you're very likely to find changes that nobody's had a chance to think about before that may be benign or might actually be very important.
So we have a lot of patients who come to us at the Lung Institute with rare genetic disease. And one of the unique programs that you're running right now is looking at rare genetic diseases and trying to find ways to intervene early to help those people the most. These are what I call orphan diseases sometimes because there's not a lot known about them, and research has not really been heavily focused on them like it has been on other diseases. Can you talk a little bit about your work in that area?
Right. So first, your premise is exactly right. The attention has been on what we call common diseases or common complex diseases, such as congestive failure and heart, early MI, all those things that kill us as adults. But there are many, many specific disorders that are caused usually by single genes or by a very simple genetic combination that can occur more early. And those individually are rare, but in aggregate, of course, there's a lot of them. So, sorry, I'm not answering your question yet. That's repeating your question. But our mission largely has been around these rare diseases as well from a research point of view. And the fundamental logic is that if you have a system that's simple enough that you can possibly understand all of the nuts and bolts that make it work, then maybe you have a chance to build up more comprehensive pictures of human molecular physiology. So, if you have two individuals and they differ by one single DNA letter, one of them is healthy and the other has a profound disease, then all of the physiology of the one with the profound disease that leads on from that one letter change is going to be informative.
Yeah, that's a brilliant way to look at it. And frequently, I think sometimes going the opposite way to look at it is going to be futile, right? Because we can't possibly look at it with just a physiology approach.
Yeah, well, we had much more hope early on at the beginning of the genome program when we realized that we were developing new genetic tools that could do comprehensive surveys of large populations. And if the genetics of even the adult common complex diseases was simple enough, then we could use those tools to find things find the genes for things like diabetes and MI.
Yeah.
But it just turns out that the genetics is more complicated, so we still don't have those answers.
But if you do find a particular defect and you want to go in and edit the genes, now with Jennifer Doudna's technology, we have genetic editing or CRISPR technology. Can you talk to me a little bit about how that's changed your approach?
Well, in terms of discovery in human populations, we're kind of doing business as usual. But once you get into the lab, once you want to go and build model systems, these new tools have opened up many new methodologies that are incredibly informative. So an example of that is this multiple analysis of variants at once. So you have now the tools where you can take a section of a gene and in the laboratory introduce every possible change in a single genetic library and put that library into a cell line and then ask which of the cells behave in which way. And then when you find the cells, for example, that grow well, then you take them, sequence them, and look and see which variants have caused them to grow faster than they normally would.
Right.
So you have now this sort of methodological link between making the variant libraries and what their function is. And that's been done in animals, of course, animal models as well, and in multicellular models and in stem cell transformation models. It's really exciting. But I think probably the most interest for everyone really is therapeutics.
Right.
And then when are we going to have gene editing that can really introduce therapeutics? And there are a few exciting examples now. And of course, the one that's at the front line is sickle cell.
Right.
And reactivation of the fetal hemoglobins. That's got it working. And I think that you see this pattern of these early, very laborious and troublesome experiments where all of the problems are unearthed, and they always cost a lot of money at a very low scale, but the problems get solved, and the process gets better. And so we can really look forward, I think, to seeing examples of where that'll be more routine therapeutics.
Tell me a little bit about the Imagine project that Baylor's conducting, and what you think that will happen.
Yeah, thanks for asking about that. We're very excited about that. So, if we agree that a good fundamental mission is to get genomics into the clinic, then we need hooks to do that. So, the unfortunate truth right now is that most physicians cannot find ready use of genetics in individuals who are acutely affected by any particular disorder. In the rare diseases, that's the exception, because that can give you more insight into the whole pathophysiology and what to expect. But in general, if you've got a sick person, you've got a sick person, you're dealing with them as a clinical problem. So the impetus to do genetic screening there is low. So we've long wrestled with the question of what could get genomics into the clinic in a way that will be readily engaged. And of course, our secret agenda is that once it's there, then our access to the data will itself enhance discovery.
Right.
So we'll build this cycle of translation, new data comes back to the lab, the discovery's made, and the data are more valuable, so there's more impetus to get it there. So I'm giving you a very long answer. But,
no, I love it.
But we've done that in pilots. And we had a pilot that is kind of our headline, our poster child, called Heart Care, which was quite limited. It was about 700 people, and it was here in the Baylor ambulatory cardiac clinics. And it was a set of genes that are known to impact cardiac health in some way, about 150 genes. And so we screened individuals, and we quite surprisingly found about 10% of them actually had mutations or alleles in genes that were essentially Mendelian conditions, these rare conditions that had simply prevailed through their lives. Now they're adults and they're finally having the effect of those genes. So this was quite exciting, and 10% hit rate is pretty good for any test in the clinic. And so we thought that's great. That'll help us get these tests into the clinic and satisfy this idea that we can do this cycle of translation and getting the data for research. But that was a pilot. And so now we've actually done a couple of other pilots and have an ongoing one here in the college. We have one in the neurology clinic with Dr. Schulman doing movement disorders. We ran one in the Rio Grande Valley looking at cardiac and diabetes-linked genes. And the hit rate for the rare alleles hasn't been quite as high. It's been more like 1%. Okay. But anyway, and so I'm still not quite answering your question. That's all the background. And the effort to do these pilots, the energy for that, the resources, very high for each one. And we're duplicating the effort. So we sat back and we said, now is the time. We have the expertise and the connections with our clinical colleagues on the front line in these pilots. Now's the time to generalise the whole endeavour. Okay. So let's start to move towards this vision that many have talked about for a long time of having frontline clinical consent that's quite general and allows blood collection, for which we can then use that from pretty much anyone who walks into any clinic, and then do generalised genetic testing, and then for those for whom it will benefit, return that genetic information that might be impactful, and then at the same time be able to use their clinical data that they will consent to being part of the research program. So Imagine is the project to do that here at Baylor College of Medicine, finally getting to answer your question.
Yeah, no, it makes sense. And when I think about commercial groups that are out there already doing this, collecting your DNA and sequencing it just for your curiosity, not really for therapeutic benefit or to try to help you understand what your genomic makeup might be, programs like 23andMe just got sold. And I think what we as patients want from our providers is to be linked to that data. That's the problem that I see right now is these commercial groups aren't linked with the provider. They aren't linked with the trusted source of someone who's caring for you or an institution that has the wherewithal to really investigate what the significance of that is. So that's why I think it's really wonderful that Baylor's doing that. Now, I know that you're partnering with Dr. Ivan Rosas. Let's talk a little bit about how your research is working in patients with lung disease specifically.
Yes, of course, he's interested in COPD, right? And it like so many conditions, it is heritable, And that is, if you have family members, primary relatives with the disorder, your risk is greatly enhanced.
Right.
And in fact, those kinds of studies could give a number that says how heritable it is and the percentage of overall risk you should be able to explain by genetics.
Right.
And then, like so many other disorders in adults, in fact, the molecular data we have only explains a fraction of that.
Yeah.
Genetics. So really the work with him is to bring about clinical translation to basically sequence his patients.
Yeah.
And to begin to find more and more of the genetic clues that lead to them, and then piece together this puzzle that will give us a better molecular understanding to fill in what's missing in our statistical understanding of what the risk is.
That makes a lot of sense. I have a good friend of mine whose mother became very ill, and I sent her to Dr. Ivan Rosas, who runs our interstitial lung disease and pulmonary fibrosis clinic. And the mother of my friend was diagnosed with advanced pulmonary fibrosis, which, as we know, sometimes can cause patients to die because it can be so advanced. And we know that the earlier you get diagnosed, perhaps the quicker you could get treatment, the more targeted therapy you might be able to get, more treatment options. And so her entire family got together. And unfortunately, this family has a history of BRCA. And the daughters had already undergone hysterectomy and breast surgery to limit their risk of getting breast cancer or ovarian cancer. So having their uterus removed, ovaries removed, breast removed, hoping not to get cancer. So that's a perfect example of where we do the genetic testing and we do something to intervene to prevent an avoidable cancer or death. So now they're asking the question, should we get sequenced? Do we need to participate in this project and get screened? And I think Dr. Rosas said, yes, let's look, let's see what your likelihood of developing this advanced lung disease might be down the road so that perhaps we can watch you and screen you closer if we see that you're at risk. Is that essentially what you plan to do?
Yes, I mean, I think that's the long-term vision.
Yeah.
And I've got to ask you, is there any physiological relationship between the breast cancer gene and all of its variable effects and pulmonary fibrosis has anybody ever made a clinical link?
I don't know. Maybe we're making it right now during the podcast.
Yeah.
I don't know. I think it's a great question. I do happen to know a few people who have both of those, but I don't know if it's just coincidence or actually a link. It might be something we can look into.
Yeah. So in these studies like the one with Dr. Rosas, we're, of course, looking at heritable mutations.
Right.
And so, we kind of hang everything around that. Now, what did you inherit from your parents? Which family members have the gene, which don't? Let's look at the differences. That's traditional genetics. But we're developing
that's like the family tree where you have the Mendelian genetics mapped out for each defect, right? So I think even I was helping my daughter do that to show color of your eyes. That's a good thing that people sometimes try to map out. And I think what we're learning now is with AI, you can do these complex maps and look at those links a little better, right?
That's absolutely true. I mean, I guess the real juice that makes things happen there is the amount of data. You need a lot of individuals, a lot of sequence, a lot of phenotype data.
Right.
And then the AI helps in those processes that bring it together and tell you what the true associations are.
So how many patients do you think we would need to have enrolled into the Imagine project before you think we get that moment of lift?
Well, it's going to be a lot for the common complex diseases if we start to select individuals not based on the phenotype, but just because they're willing to participate.
Right.
So ultimately, we're looking at hundreds of thousands.
Yeah.
And of course, it's not practical right now to do. I mean, it's simply too costly. But if we begin on that journey, then we're moving towards this vision of basically everyone having their DNA sequence in their medical record, because it might be useful in some context. And then with that behind us, then we can start to reach into these very large data sets, use these tools that you're alluding to, and see the associations that can help to give us some of these genetic answers.
This is great. I feel like you're one of the few individuals in science that has a little bit of a crystal ball. You see the future a little bit more clearly than the rest of us do.
Well, I think not being encumbered by the daily reality of the challenges in the clinic is something that allows people to be futuristic. And of course, usually what happens is we make some developments, do some tests, and start to talk to our clinical colleagues, and very quickly we're brought back to earth.
Right.
Based on the realities of what's in the clinic. So this is so valuable, and what's great about our environment, I think, is that we can have these kind of conversations and do things and then shape them back to reality and usefulness.
Yeah, I do believe that. I think one of the strongest things of being at Baylor College of Medicine is we have PhD scientists who are dedicated to the investigational work. We have clinicians who are dedicated to the clinical work, and then we have those rare individuals who can move between the worlds and talk between the worlds. And I think it's very important for us to stay connected to one another and stay relevant so that the scientists are doing research that translates into real actionable change in our clinical practice. It's definitely my main goal as the executive director of the Lung Institute to try to keep us focused on the research projects that we're funding and the type of science that we're looking at, and highlighting that it stays relevant to our patients, keeping our patients in the center.
Well, here, I say. This is
Yes.
music to me, and, we're there for you.
Great. Well, thank you so much for joining us. It's just a true honor to get to sit down with you, and we're hoping to have some more podcasts with you to really continue to learn about your work, continue to learn about what you see happening in the future, and to continue to link our patients with you so that they can learn about the Imagine Project, your research, and ways to help even further that research, I think, with grateful patients and benefactors to help support this research as we look for unique sources. So,
Well, the pleasure's mine to be here, and please keep on your great work.
great. Thank you so much, Dr. Gibbs. This is Shanda Blackmon bringing you the On Air podcast for Baylor College of Medicine Lung Institute. Thank you so much for joining us.